All right, let's make a protein version of it. I'm going to create another function, run protein quality control. Again, we're going to pass RNA and protein to it. And I'm going to make another file. Let's say protein distributions. And let's define the function in the module. This is gonna probably give us another tuple of and data and another annotated data. So from anno data import and data. And also maybe here. These are software engineering standards. It's good to have it, even if you're just researching. So in case things go into production, you have it easier. Production ready code. Ideally, you want every kind of action you make to be put in a separate function, and then you call functions within each other. But yeah, so I might end up doing that all, but for now let's focus on the. And by the way, here I guess we have to pass protein real. And here as well, because these are the ones that didn't have negative controls, right? Some cells may have suspiciously low protein signal. If the protein target is mostly technical noise, the model will struggle no matter how good the algorithm is. So we want to inspect the total protein counts per cell and how many proteins are detected. Like how many proteins have non-zero expressions in each cell. So I'm going to define total protein counts to be, also we gotta import numpy. So I want to sum protein counts across all proteins for each cell. So X is the expression matrix, right? So we're going to sum over all the proteins for each cell, right? we want to flatten this into one dimension. So per each cell, we're going to have the number of proteins stored in this variable. Now, I'm going to create another variable number of proteins detected. And I just want to check wherever the expressions are non-zero, and then sum them for each cell. Again, we flatten it to one-dimensional array. So how many proteins have non-zero expression in each cell? Or how many proteins were detected per cell? Right, I might have missed. Okay, yeah. This is gonna look fine. Now we'll get a summary printed. So the only thing that I'm seeing here that are kind of a little needs attention is that total protein counts has a mean of 3,000, but then we have the maximum to be 30,000. This means a small number of cells have unusually high total antibody signal. So this could be something we can, investigate further. So let's plot a histogram of total protein counts per cell and see it. So and I'm gonna go protein OBS total protein counts dot hist bins, let's say 50. We're gonna label x-axis to be total protein counts per cell, and then the y-axis will be number of cells. And the title could be distribution of total protein counts. And I'm going to save this. give it DPI 300. And we're gonna close it. Also, I wanna put this inside git ignore. So let's go ahead and debug. Oh, I think this must be matplotlib.pyplot. Yeah. All right, so the distribution is kind of right skewed with a clean peak right here. It's probably around between 2500 to 3500. So the left's tail, somewhere between 0 and 1000, have less than 200 cells. About 200-ish cells have very low protein counts. These are kind of suspicious, worth cross-checking. But the right tail is kind of clean and short. Unlike the RNA data, there is no extreme high count tail stretching to like 50,000. So everything is kind of dying around 10,000. This means antibody aggregation is not a real problem here. But as you see, the x-axis goes up to 30,000, as I said before. And this is important. It kind of means the plot was auto-scaled to match other plots or future plots. But the actual data range is much narrower. It kind of dies off here. So no cells are in the between like 10,000 and 30,000 range at all. But this part is still a little weird, and I would want to go back and define a minimum protein counts to be, let's say, 1,000. And I'm gonna remove the proteins that have anything less than that. Yep, and then I will probably change RNA based on these proteins as well. So if I go up here and try to write something here, so this function checks basic protein quality control metrics. And now I want to look at the distribution of each protein target. This is important for ML because if some proteins are highly expressed in many cells, expressed only in one immune cell type, mostly zero, noisy, it's gonna be impossible to predict from RNA alone. Total protein counts and number of uniquely detected proteins per cell. Okay, so let's add this. Add RNA quality control, filtering counts unique proteins per cell. So now I'm going to define a new function. Plot raw and log distributions. Let's do this for now. We're going to change it later. So we want to look at every protein separately. And to get a clean statistical summary of every protein in one shot, there is this Panda feature called Describe. And I would like to just convert this protein that we have, which is an AND-data object, this sparse matrix X to a Panda data frame. But before that, I would have to first convert it into an array. And so toArray also is a method that only exists in sparse matrices. So I'm going to check if our proteins have X is at all a sparse matrix. All right. Else, just take the matrix as it is, right? So I want to make a data frame out of this. I'm going to call it protein df. Okay. But also I want the index to be proteins and then columns to be variables. Okay, and now after this, we're going to see protein df dot describe. Perfect. And so if we want to see proteins as rows and statistics as columns, we want to transpose this, right? Let's see what is gonna show. So if we're looking for some red flags here, this is going to be the median, right? And we want to see what cells have the most difference between mean and median one values. So if I look at, for example, CD11B, mean is 710 and then median is 19. So this is a very large difference. That will make the plot to be skewed to the right? So to get around this, we will probably need to use log normalization. Now, if we look at standard deviations, difference, and mean, the ones that have very higher standard deviation versus the means, such as again CD11B. So we have 1134 and 710, or I think almost really most of them, or if not all. But if standard deviation is higher than mean, it means this specific protein is not uniformly expressed. It is basically off in most cells and on in very few cells. So like very high values for some cells and very low values on most of the cells. To get around this, besides doing the log transformation, we should probably do something called centered log ratio, if you're not already familiar. But yeah, so if we don't fix this, then any model will treat the high cell as extreme outliers, and the near zero cells all look identical to it. So no resolution between them. But there are ways to fix that. And lastly, if we compare the maximum with 75th percentile, if again there is a huge gap between these two numbers, then this is an outlier, right? And so log transformation will spread them out evenly. And also, if you see like almost all of them except for two are having a minimum of 0, that validates the sparse matrix too, besides the point. But yeah, that's all we can see from here. So we can go ahead and try to plot this. Give it a 16 and 12. Yeah, this Y will move the title slightly upward. I'm going to do adjust spacing so plots do not overlap. Type layout. Maybe let's make a new folder. Dist P raw protein counts. And so I'm gonna apply the log transformation. So this will apply log 1 plus x transformation. And again, I'm going to make another plot so we can see it. So maybe we want to return the logged version. Okay. Now let's go ahead and try to run. Oh, this has been not assigned. Wait, okay, this is subtitle. Now, when we look at the logged version of the data, we kind of see that there are little bumps here. So this guy has 2, this has 3, and then there are some of them that has technically nothing. Interesting. So these 2 humps plots are called bimodal distributions. In this case, it probably means protein has dynamic range and it varies meaningfully across cells. And so the model has actually something to learn, like an RNA pattern that predict low versus high. So you get kind of a range, you know. But for here, for example, CD34, almost every cell values like 0 to 1. So there's nothing to predict, right? Every cell is the same. The model learns nothing really useful. If we have more than 2 bumps, such as this guy or this guy, it means there are more biological complexities. And so it's more informative, and the model could learn like a more complex pattern in case of a regression. Just from looking at the shapes, we now know that these bimodal shapes are the proteins that worth predicting. All right, so I'm gonna go back and maybe we can take this guy out and define a new function called convert and to data frame. So we're going to do this to kind of like make this production ready code. So this function looks at the distribution of proteins individually. Alright, so let's make a commit. Again, from a series of quality control, distribution, analysis, and log transformation. Now we're going to move on to check protein sparsity. Just to know for each protein, check how many cells have 0 values. We're going to do that in the next video. Thanks for watching and hope you enjoy this.